For those of you who don't know who I am, Alki Soldas. I'm currently Dr. Wang and Dr. Knight's fellow in rhinology. I'm presenting the work that I did during my PhD a few years after my PhD in Adelaide. But I'd also like to credit Dr. Wang and Dr. Knight for their great mentorship during this fellowship, which really has been a life-changing experience in many ways. To give you a bit of a background as to where I'm from, this is Adelaide, or Adelaide in Greek or German pronunciation, as I just found out. It's a town of about 1.5 million people. It's the fourth largest capital city in Australia after Sydney, Melbourne and Brisbane. Perth is soon overtaking Adelaide in population-wise. It's the slowest growing capital city and it's the oldest population. Another interesting fact is that it's the only capital city that wasn't settled by convicts. So when the English shipped all of their prisoners out to Australia, all the governors and all the aristocrats moved to Adelaide and took up their residency there. And so if you are familiar with the Australian accent, people from Adelaide actually speak more like the English than the rest of Australians, but not to the same extent as the English, obviously. So the university where I'm affiliated with is Adelaide University. It's centred, as you can see, marked out in the diagram across the River Torrens. And the Royal Adelaide Hospital, which is an 800-bed tertiary hospital, is located in close proximity. Now that's the current Royal Adelaide Hospital, which, as you can see, is quite run down. This is going to be the hospital for 4 years' time. A bit like Stanford, we've got one of those trajectory sort of structures. So Adelaide does have some famous people. So this is Howard Florey, who went to Adelaide University and was one of the first graduates from Adelaide University Medical School. And for those of you who are familiar with Tennessee, I'm sure we all are. The Bragg father and son were the only two physicists to be awarded the Nobel Prize in the father and son category. And they worked together on and together they won the Nobel Prize in Physics. We have our contribution to that. Although not too many people know about us, we do have a few important people that have come out of Adelaide. So the only financial decision today what I want to present to you are essentially the key features of this cycle. I want to show you how in our laboratory and our centre we went from determining that biofilms were important in chronic rhinosinusitis to characterising them, to making the link between the actual structure and the pathogenicity involved in chronic rhinosinusitis, showing how they're clinically relevant to our disease, and finally developing or starting to develop treatments against it. And in the next 45 minutes or so, I wish to try to progress around this circle to show you our 3 to 4 year research that has come full circle in trying to better elaborate the role of biofilms in chronic rhinosinusitis. So what most of us know about bacteriology, and what I knew about bacteriology before I embarked on my PhD, was really the work from Robert Koch. Now, Robert Koch was a German physician who developed these postulates. And these postulates were that in order to prove bacterial causation of disease, you needed to isolate the bacteria from an infected animal or organism. You then needed to grow it out, and then needed to inject it into a healthy organism and show replication of the disease. And so you really needed all of those features to be fulfilled in order to say that a particular bacteria caused the disease. Now, this was a problem for many chronic diseases because often you can't isolate bacteria, or often there isn't a suitable animal model that's permissible to the disease. And as a consequence, for many years, a lot of chronic diseases have been suggested to have an inflammatory component because the bacteria can't be isolated or an animal model can't be replicated. And this is kind of what happened with chronic rhinosinusitis 20 to 30 years ago. Although bacteria were thought to be an important role in acute rhinosinusitis, their role in chronic rhinosinusitis became less important, and we couldn't prove bacteria were always present. What we now know is that form that Robert Koch described, that form that we grew up with as medical students and played out in the petri dishes, really only describes 1% of all bacterial forms. That's a planktonic form. That's a free-floating form that we can culture out into a petri dish. But in actual fact, 99% of bacteria, the same bacterial species, adopt a more resilient form called a biofilm. Now, biofilms aren't a new concept. Biofilms were first described by a Dutch lens maker back in the 16th century. He wasn't a scientist, he wasn't a doctor. He wrote multiple letters to the Royal College of Surgeons in England, but because of his status, those letters were ignored. What he did was he used his high-power lenses that he created to study his environment and his surroundings. He took scrapings of homeless itinerant people and looked under these lenses and commented on what he found. And what he described were the two forms. He saw these motile forms that rapidly spurted across his visual field, and then he described these much more common clusters that seemed to rotate around themselves and not change in motion. And so he proposed that there were two bacterial forms that existed. But unfortunately, as I said, his theories and his observations were ignored until about 30 years ago. So what are bacterial biofilms? Well, bacterial biofilms, and this is a scanning electron microscopy picture of a bacterial biofilm, are structures that are formed primarily of microorganisms. These microorganisms can be bacteria, they can be fungi, they can be protozoa. So these organisms attach themselves irreversibly to a surface and encase themselves in a self-produced exopolysaccharide matrix. And it's this matrix, which is formed primarily of water, but also contains polysaccharides, nucleic acids, and proteins, that protects the biofilms from attack from the external environment and from the host itself. So these are the important structural features that make up a biofilm. But there are also important functional features. The bacteria that exist within biofilms have been shown to transcribe up to 70 different genes. As a consequence, they have a different phenotype to the planktonic bacteria, despite being the same bacteria themselves. They have a much slower growth rate. forms. And those planktonic forms then embolize into the system. And so this is a very important example of what we see in our patients. We're often told to culture our patients when they have fevers. And the reason for this is when a biofilm is embolizing. This is when a biofilm has dedifferentiated into a planktonic form and can now be detected by the body, and the body can mount a response to it. So when we have patients with central line catheters or that's where bacteria have dedifferentiated. So all of the both the structural and the functional features make biofilms a very attractive and plausible explanation for chronic diseases. In 1999, the Centre of Disease Control in the United States estimated that up to 65% of all human bacterial infections were driven by biofilms. It's now thought that up to 80 to 90% of all bacterial infections are biofilm driven. And you can see from the list here, there's many of the common things that we come into contact with every day. So what about in ear, nose and throat? Well, almost every aspect of ENT can have biofilms involved. From the prosthetic ventilation tubes we put in our patients, to the tracheostomy tubes that we cannulate our airway patients with. Otitis media with effusion, although once considered to be a sterile effusion, has now been proven conclusively to have biofilms present within them. Cochlear implants, cholesteatoma, even the chronic tonsillitis patients have all shown to have biofilms. So you can see that they're really an important aspect of all ear, nose and throat disease. So now I'd like to progress on to their relevance to chronic rhinosinusitis. Well, there have been a whole host of studies, the first published in 2004, which tried to look at the prevalence of biofilms. The early showed that biofilms probably weren't as important in chronic rhinosinusitis patients or as prevalent in chronic rhinosinusitis patients as was previously postulated. Now, as a background to that paper, we first wanted to compare the three different imaging modalities to image biofilms. We compared using the scanning electron microscope, the transmission electron microscope, and the confocal scanning laser microscope. All three have very different ways in which you prepare the tissue, and all three have very different ways in which you can image the biofilm. The scanning and the transmission electron microscopy involve the use of dehydrating agents such as aldehydes and alcohols, which can distort the structure that you're trying to image. In addition to that, they can't differentiate between mucus and bacterial biofilms because both contain long-chain polysaccharides in their structure. So in order to test this, we developed a sheep model in which we looked at 24 merino cross sheep with 48 sinuses. We had 4 treatment arms. We had one where we didn't do anything at all. We left the ostium of the frontal sinus open and didn't put any bacteria in, and that were the frontal sinuses. We then opened we left one of the frontal sinuses open but injected bacteria to see what would happen if you didn't have the anatomical obstruction that we often see in sinusitis patients. In another group, we simply obstructed the ostia, and in the final group, we had a double active arm of what we tried to mimic chronic rhinosinusitis with. The arm where we obstructed the ostium and we injected bacteria. All the sheep underwent a standardised endoscopic sinus surgical procedure using similar equipment to what we use in the OR, just extended in their nature. And then we grew biofilms out to 7 days because we determined in vitro that it took 7 days for a biofilm to reach a plateau phase. At day 8, we harvested the mucosa from the frontal sinus and then we subjected it to 3 imaging modalities. And what you can see side by side here are the intraoperative and the CT scan of what we did. So the first step was to remove the middle turbinate of the sheep. And you can see the turbinates look very similar to what we see in our humans, although they're a lot longer and they take up a lot larger part of the nasal cavity. Following removal of the turbinate, we cauterized the stump, and then we proceeded to resect the ethmoid air complex. Here you can see us putting a pledget in to provide hemostasis. And what you'll see is us now removing the ethmoid complex or ethmoid terminal with blade sleeves and through cutting instruments. Following this, we wanted to make sure that we had actually isolated the frontal sinus, so we performed a mini trephination through the external part of the sheep's frontal table, and then we injected fluorescein through the frontal sinus trephine to confirm that in actual fact we were going to be blocking the right sinus. We then placed cotton wool into the ostium. It was a sterile cotton wool pledget, and we extracted the ostium and then injected bacteria through the trephines. So that was our animal model. What we then did was we then harvested the mucosa, and you can see us here removing the frontal table and then resecting the mucosa. The sinus is very variegated, so you have lots of nooks and crannies to the sinus. And here you can see what the sinus looks like once it's been harvested. We excluded the part of the sinus that was in contact with the cotton wool pledget because that was likely to have a foreign body reaction. And we only obviously analysed the bit that was distal to that. And what we found was this is what an acutely sinusitic sinus looked like compared to a normal sinus in the control group. So you can really see that we were creating a relative degree of inflammation. What we found was that when we looked for macroscopic evidence of sinusitis, the only sinuses in which we consistently created this inflammation was in the sinuses that we injected bacteria and obstructed the ostium. When we looked at the different imaging modalities, we found that the scanning electron microscopy tended to overcall these biofilm structures. Even in the control group where there was no macroscopic evidence of sinusitis, we were seeing these three-dimensional structures that had been described in the literature by previous authors to be biofilms. The transmission electron microscopy seemed to have more specificity, but it lacked sensitivity in terms of detecting these biofilm structures. What we found was using this confocal scanning laser microscope, we did have identical correlation to what macroscopically and microscopically we were seeing. And there are reasons for this, and I'll show you very quickly. As you can see on the control of the biofilm group in the top left-hand picture, that's a scanning electron microscopy picture. Mucus and biofilms look identical because, as I said, they both contain these long-chain polysaccharides. In the transmission electron microscopy group, we can see that the control group is very easy to differentiate from the biofilm group, which contain the bacteria. But because you're taking ultra-thin sectioning, the chance of you missing a biofilm is much higher. Now, with the confocal scanning laser microscopy, what we could find that we could easily determine the difference between a biofilm and a control. Here you can see the large nuclei of the epitheliated epithelial cells, and here you can see the small 1 micron in diameter bacterial clusters interspersed amongst dead nuclei of the epithelial cells. And the advantage of this was we were also able to look at the tissue in a much larger degree. We weren't having to fix the tissue and use ultra-thin sectioning. And also we could scan through the whole specimen to see the depth and the thickness of these biofilms. So we then took this to our patients to determine the prevalence of biofilms in our patients. We had 38 patients who were consecutively enrolled with sinusitis, and we used the sphenoid mucosa from transsphenoidal pituitary patients who had no macroscopic or radiological evidence of sinusitis as a control group. We collected all the relevant data, and then we subjected them to confocal scanning laser microscopy and showed that in our control patients, we saw healthy epitheliated cells. In our biofilm patients, or in our chronic rhinosinusitis patients, in about 44% of patients, we saw these three-dimensional structures called biofilms. So we concluded from that perhaps, at least in our cohort, the rate of biofilm detection was much lower than previously had been quoted in the literature. So we've now said that we have biofilms in sinusitis, but what makes up the biofilms? What are the key organisms that are involved in biofilms? And so that's what I'm going to speak about now. So to do this, we used fluorescent in situ hybridization, which allows you to use specially marked nucleic acid probes to target specific bacteria. And we tested for the common pathogens in chronic rhinosinusitis: Pseudomonas, Staph aureus, Haemophilus, and fungal probes. And what we found was that in this particular study, 72% had biofilms. And I'll go into the reason for the slightly higher figure than we had quoted previously. And we had no biofilms in our controls. We found that again, Staph aureus was present in 50%. It was the most common. And this probably is that what we were seeing in a confocal scanning laser microscope were the robust big biofilms of Staph aureus. We found in this experiment that the Haemophilus influenza biofilms were much harder to detect because they were a lot finer, they were a lot less structured, and they were a lot less populated with bacteria. We also found that they were present, but in a much lower rate. In 28% of our patients, we had Haemophilus influenza, and in 22% we had Pseudomonas. And we also found that these biofilms tended to have lots of bacteria comprising them. 19 of the 50 had more than one species, and 6 of the 50 had more than 3 species. Interestingly, this was the first paper that was published on fungal biofilms of chronic rhinosinusitis, and we found that 20% of our patients actually had these fungal biofilms as well. The interesting thing was that we found that there was a symbiosis between fungal and Staph aureus biofilms, so that 64% of the patients that had fungal biofilms also had Staph aureus. So to see this relationship, we then created another animal model to see the relationship between fungus and Staph aureus. We used intraperitoneal sensitization of the abdomen to sensitize the sheep to fungus. And then we inoculated their frontal sinuses with fungi to see how many would form biofilms. And when we inoculated these sheep with biofilms, we found that only 10% of these sheep that were inoculated only with fungus ended up forming fungal biofilms. However, 80% of the sheep that had fungus and Staph aureus inoculated together formed biofilms. So Staph aureus seems to be a very important cofactor to the development of fungal biofilms. And we postulate the reason for this is that Staph aureus, being a very virulent organism, can get in there, it can denude the epithelium, and it can create an environment that is really important for fungus to grow and to form a biofilm. So what about the pathogenicity? So we know that biofilms exist, but are they a cause or effect of chronic rhinosinusitis? Well, in order to do this, we've only made sort of baby steps into determining the pathogenic link. But we have made some progress, and I'll present it here. Before I go into that, I want to speak a little bit about superantigens. Now, about 6 or 7 years ago, superantigens were the buzz term. It was thought that 50% of patients with polyposis had superantigens present. Since then, further evidence led to the importance of superantigens through the detection of immunoglobulins against these superantigens. The fact that we showed a specific expansion of specific T lymphocytes with the domains existent in superantigens. And finally, when we hit in vitro tissue with those superantigens, we generated a very similar T helper 2 cytokine induction that we saw in our patients with polyposis. Now, the fact that we're seeing superantigens in 50% of CRS patients and we're seeing biofilms in 50% of CRS patients, we thought that perhaps the two may be linked. So to further investigate this, we looked at 33 patients with chronic sinusitis and polyps, 20 patients without polyps but sinusitis, and then 15 controls. And we tested them for the presence of Staph aureus biofilm using the FISH protocol, and again we found about 50%. And then we tested them for the presence of superantigens. We found 52%, but we only found the 52% in the polyposis group. Although we did find biofilms in the non-polyposis group, there was no superantigens present there. We ran a statistical analysis, and what we did find that there was a link between the presence of biofilms and superantigens, which was statistically significant. But this link was low. And so we postulated that perhaps superantigens were a way that biofilms were mediating their pathogenic influence on the body, but wasn't the only way that it was being done. So to investigate this further, and this does get a little bit basic science heavy, and it will only be brief, we did some cytokine assays to see whether or not the cytokines that the biofilms were producing were similar to the cytokines that are seen in a superantigen response. We used a special technology called Luminex technology, which allows you to screen through a whole host of different inflammatory cytokines. And what we found was that interleukin-5 and extracellular protein were the most important inflammatory mediators that biofilms tended to be associated with. For the patients that didn't have interleukin-5 present, all had TGF-beta present. So when put together, the three of them had 100% predicted value of biofilm being present. We then ran the same screen on our Staph superantigen patients and found a totally different cytokine pathway. We found that total IgE was up and myeloperoxidase was up. And so that made us think that perhaps the two aren't as intimately linked as we had initially thought, because they're working through two different pathways. To further clarify this, we then predominant side which sit down here. The cytokine expression profile tended to exist in this halfway between the T helper 2 and the T helper 1 area. When we had superangens present and biofilms absent, they had a different cytokine expression. The most important finding of this, and I suppose a takeaway point from this complex graph, is that when you have superangens present and biofilms present, the cytokine expression was completely different to the other two, further giving evidence that perhaps the two are synergistic rather than codependent on each other. So the way that biofilms and superangens are affecting the immune system or the patient with chronic rhinosinusitis are in independent but synergistic pathways rather than being dependent on each other. We published two papers to see what biofilms and both in our retrospective study and in our present tended to have worse symptoms and continue to have worse similar picture was seen that up to 12 to 18 months from the time of surgery, these patients still had worse endoscopy scores than the patients who were biofilms. Does that actually affect the outcome? So can we differentiate the biofilms that patients have on the outcomes? And so to sort of look at this in more detail, we looked at 24 patients. We ran our FISH probes of staph, Haemophilus, Pseudomonas, and fungus again. And then we looked at their clinical, radiological, endoscopic, and culture results to try to work out whether or not particular biofilms had worse prognosis, or whether we could lump all biofilms together. What we found was that our two most common biofilms again were Haemophilus and Staph aureus. But we found a completely different phenotype of the Haemophilus and the Staph aureus biofilms. The Haemophilus, which were much less robust biofilms on the FISH demonstration, actually were less symptomatic. They had less severe radiological disease, they had a more rapid disease resolution, and they had less post-operative infections than the Staph aureus biofilms. When we represent this graphically, you'll see in a minute how impressive it is. The Staph aureus biofilms were more symptomatic. They had more severe radiological disease, and they tended to have more persistent disease post-sinus surgery. So if we look at the sort of the plot of post-operative outcomes, we can see that these Haemophilus influenza patients, although they were bad initially, tended to resolve pretty quickly, whereas the Staph aureus biofilms tended to have a worse protracted clinical outcome. So just because a patient has a biofilm doesn't mean that they're predisposed to a bad outcome, but if they have a Staph aureus biofilm, it does put them in that worse disease category. So what about treatment of biofilms? Well, we know that biofilms are resistant to antibiotics, and it's thought they're resistant by four main mechanisms. First of all, biofilms are associated with a charge. The biofilm charge is negative, and so it tends to repel antibodies that are positively charged, such as aminoglycosides. A second mechanism is that biofilms may contain enzymes that deactivate antibodies, and Klebsiella has known to contain beta-lactamase, which deactivates ampicillin. Thirdly, it's thought that bacteria within biofilms have a resistant phenotype they have nutrient deprivation, and as a consequence, they shut down their growth and metabolic rate. So they're not responsive to the antibiotics that manage to get deep within the sort of depths of the biofilm. And finally, deep within these biofilms, we have highly different milieus. We have acidic, anaerobic, hypoxic milieus, and they're each different. So although you might be able to treat 90% of the biofilm, 10% of the biofilm may remain resistant and may form a nidus that can repopulate the entire biofilm. So if we want to target a biofilm, we really need to target it at three aspects. We need an antibody, obviously, to kill the bacteria. But before we have the antibiotic being effective, we need to physically be able to remove the biofilm. And this is why mupirocin, I suppose, is one of the antibiotics that we tested. Now, mupirocin is isolated from Pseudomonas fluorescens. We use it commonly in our rhinological patients. It's stable in nasal secretions, it has very limited nasal absorption, and when it is absorbed, it's rapidly cleared and inactivated, so it's a safe antibiotic to use. Also, it belongs to a completely different class to all the antibiotics we use, so there's very little chance of developing cross-resistance if you become resistant to one antibody. What we did was we tested this out on our clinical isolates. So we took patients with biofilms, and we basically took the Staph aureus from their nose and we grew them out onto petri dishes. Now, all of these clinical isolates, when they were grown out in the planktonic phase, were sensitive to vancomycin, ciprofloxacin, and the mupirocin. But what happens when these planktonic bacteria were then grown into the biofilm phase? What we found was that although all of these planktonic bacteria were sensitive to these antibiotics, the biofilm bacteria were only really sensitive to mupirocin. In order to be able to kill those bacteria using doses, we had to use supraphysiological doses of ciprofloxacin and vancomycin, doses which are far too toxic to use in humans to kill the bacteria at the same level that the mupirocin was. So this gave us good evidence that mupirocin was probably the most effective irrigation to be using in our patients. We then took this back to our sheep model, and in addition we tried some other antibiofilm agents. We tried gallium, which is an iron chelator, and we tried a surfactant produced by Medtronic called citrionic surfactant in conjunction with a hydrodebrider. And the thought was that this surfactant would go in there, it would disperse the biofilm, and then the hydrodebrider would go in there and be able to de-agitate it and remove it from the surface. What we found in our biofilms grown for 8 days, that when we harvested them immediately post-treatment, day one post-treatment, all treatments were pretty effective compared to saline. But in the group that we let the biofilms repopulate and reharvest after 8 days, we found that only mupirocin had the prolonged antibiofilm action. So basically, all treatments are effective in the acute phase, but they don't get rid of the biofilm to the same extent that mupirocin does, and they allow for repopulation to occur. And that's why a lot of these agents, although touted to be antibiofilm agents, really need to be tested long term. We tested mupirocin out in 16 of our refractory CRS patients who were refractory to every other medical treatment that we'd offered, and we found that in these patients, 15 of the 16 had improvements in their SNOT-20 scores, their endoscopy scores, and the symptom scores at 3 weeks of mupirocin irrigation. The unfortunate thing is that 3 months later, when we stopped the mupirocin, 70% of the patients had regrown the Staph aureus. So mupirocin is great while it's being used, but you do get a repopulation eventually down the track. We then did a randomized double-blind placebo-controlled trial to see whether or not mupirocin was more effective than conventional oral antibiotics. And so in this trial, we used mupirocin irrigations with the placebo tablet, or augmented with a placebo irrigation. We had 25 patients and we randomized them to a 28-day treatment. That was a treatment kits that I described to you. And what we found that immediately in the post-operative group, the mupirocin group had a higher rate I suppose this is the important thing that I was highlighting before. This improvement really was immediately. 3 months down the track, although it was still decreased, it was no longer statistically significant. So once we stop using mucurus in our patients, there's a high rate of relapse, which is what we do see in our clinical practice in the sinus clinic downstairs as well. So this brings me on to the final part of my discussion, which is about the use of honey. Now, honey has been touted to be an antibacterial agent for years and centuries. As far as Hippocrates in 400 BC and the Ayurveda from Indian practice in 2000 BC. And the reason why honey is thought to have its antibiotic or antibacterial effect is through osmosis, through its hydrogen peroxide content, through the fact that it's highly acidic and the fact that it contains natural antioxidants. What I want to focus on today is a special antibacterial agent called methylglycol, which is found in certain aspects of honey. Manuka honey, exactly. So Manuka bush is found only in New Zealand, and there's a unique Manuka factor which is thought to provide this particular honey with antibacterial properties. And this particular property is called methylglycol. Now, if we look at our regular, and in Australia, our table honey is called Capilano honey. If we compare our table honey to honey, we simply put Manuka. In another group, we treated them with the table honey. Another group, we and then we created super Manuka honey by augmenting the methylglycol on top of the Manuka honey. And then we also created super table honey by adding more methylglycol. We then grew our biofilms for 24 hours, and we tried to determine what the important concentration was to achieve biosodality. And what we found was that just above 0.53 milligrams per mil of methylglycol was a concentration that you needed in order to kill bacteria. Down to a much lower level that would be tolerated. Now, there is a problem with Manuka honey. When you get to such methylglycol, it's thought that you can start to get superoxide formation, and you can actually start to get death of cells. So it's thought that you can actually induce cancer and diabetes in cells by using super-augmented rates of methylglycol. So that's why Manuka honey really hasn't made it into the market yet, because the research shows that it's probably not safety proven yet to be useful in our patients. So I suppose in conclusion of the biofilm treatments, it is promising. Honey does have this anti-biofilm agent called methylglycol, and that we can augment the amount by adding more methylglycol, but we really need to be careful with the amount that we give. So any questions? It's a lot of work to cover in 45 minutes, but it probably summarises the 4 or 5 years of the research that we did before I came here.
I call that the in a writing commentary, and he mentioned Manuka honey. His daughter had terrible eczema, and she became allergic to the steroids. So he was using Manuka in the tube form, in the cleared right up. I bought a couple. This was over the counter in the pharmacy. Brought it back to the States. Had a couple of patients who were sensitive to steroids that had eczema. And anecdotally, it cleared right up. And one lady's been 6 years now without any problems. But she puts it in the ear. Now I'm worried because you're telling me that it may be carcinogenic.
Yeah. Diabetic reports have its useful as well. Again, it's anecdotal, but some people have had
getting good enough results just treating staph, assuming that it's that issue of bioinfections.
So my only worry is that it's a really good agent. It's obviously good at decontaminating the body from Staph aureus as well when they use it. I mean, it's such a good agent that in my patient, in the patients we see, I think it's probably very treatment. When we started this study, we were allowed to send patients to a compounding pharmacy to get the Bacterial gel to dissolve it in. We're no longer allowed to do that in Australia. It's under TGA, which is your FDA equivalent investigation, because they don't want to lose it for the treatment of orthopedic infections and so forth. So, in Australia is very vigilant on their resistance to antibiotics. So I'm not sure. I don't know. I think we've got to develop a better treatment that's not antibiotic based.
Question I had was just about your population down in Adelaide and ours in North America. It seems like PJ, you're the guys down there. The team just had to see such a just recalcitrant and stubborn form of sinusitis with these thick biofilms, and I don't think we've seen them as much or to this level. Is anyone? What's the nearest correlate in North America in terms of studies that? Do people find the same percentages of staph biofilms in the U.S. and Canada? Or do you think it is something about
so I did a
your
study
population?
on contamination of irrigation bottles. And the interesting thing was that in North America, Pseudomonas is the most commonly isolated bacteria from your irrigation bottles, whereas in Australia, it's Staph aureus. And we hardly pick up any Pseudomonas. And I'm not sure if that's our culture techniques or if it's the fact that we have a different population of bacteria. But I can say that just from sort of personal experience, when we get patients that come back who are resistant to treatment, they're a lot worse than what we see here. They just, whatever you do, they just won't get better. You do drill outs and they have this thick pus coming out of their sinuses. And I think we might have super staph in Australia for some reason, and I don't know why. But I think the biofilms are very different. I really do.
Is there
It could be a high rate of allergic fungal sinusitis as well. It could be the symbiosis. We have a much higher rate of fungal sinusitis than you do here. For sure.
gets a little frustrating.
So that's my Manuka.
Very good.
It makes it to me